Hello one and all, welcome back to the last video in our series on the nervous system in chapter 12. So the last thing that we need to discuss now that we've talked about neurons, graded potentials, action potentials, and all that, once the action potential reaches the end of the axon, what happens then? So let's get into that. So once the action potential does reach the end of the axon, which we call the axon terminal, the signal ends there. We cannot conduct the action potential any further than that. So because there is a very small space in between the axon terminal of this cell that you see right here and the cell body of the next cell in line, the cell that will be receiving the signal, because those two cells are not connected in any way, they're not touching each other, their cytoplasms are not connected, we cannot transmit an electrical signal any further. For that reason, we are going to get clever and we are going to convert our electrical signal into a chemical one. The chemical signal in this case is a type of messenger molecule called a neurotransmitter. So to get some nomenclature out of the way, since one neuron is always going to be signaling to another cell, that cell could be another neuron, or it could be a muscle fiber, or a glandular cell, we're going to need some nomenclature to know which cell we are referring to. The cell that is doing the signaling, meaning the action potential reaches the axon of this particular cell, we call this the presynaptic neuron. So the whole area in which the neurotransmitter acts is called the synapse. So therefore, the cell that exists before the synapse is called the presynaptic cell. The cell that is going to be receiving the neurotransmitter signal is called the postsynaptic cell. So release of neurotransmitter from the presynaptic cell into this very narrow space, which is called the synaptic cleft, is going to cause the opening of ligand-gated ion channels on the postsynaptic membrane, and this will stimulate a graded potential. So we kind of start over in the postsynaptic cell based on our previous discussions. So first, let's talk about how neurotransmitter gets released once the action potential reaches the axon terminal of the presynaptic cell. So once the action potential reaches this area, we are going to open a new type of voltage-gated channel. These are called voltage-gated calcium channels. So calcium, because it is at a very low concentration inside this cell and a relatively high concentration outside, this will cause calcium to follow its concentration gradient and rush into the inside of the axon, into the axoplasm. Now, what you might be thinking here is, if you're thinking a couple steps ahead, you might be thinking, okay, calcium, it's a positively charged ion, it's going into the cell, so therefore we're going to get depolarization. I like the way you're thinking, but unfortunately for you, the depolarization aspect doesn't really matter here. Calcium is going to act as a second messenger here. So all this calcium that is now in the axoplasm is going to bind to proteins that are located on these synaptic vesicles that store up a very vast amount of neurotransmitter, and this is going to act as the trigger we need to release these vesicles by exocytosis. So without these voltage-gated calcium channels, all this neurotransmitter that is stored up in the axon terminal would never be released. But once those calcium channels open, we can then release all this neurotransmitter into the synaptic cleft, and then it is available to bind to ligand-gated ion channels that exist on the membrane of the postsynaptic neuron. So this is one of those slides that I recommend you bookmark because there's a lot of good information that is all summarized here in one place. So you see it broken down from the presynaptic neuron to the postsynaptic cell or neuron. They call it neuron here, but it could very well be a muscle fiber or something else as well. So just a reminder here, the presynaptic neuron in here, even though it starts, chronologically speaking, they're saying it starts with an action potential. Just want to make sure that you remember that action potential only happened because this presynaptic neuron received an EPSP of its own that passed threshold at the axon hillock. So every action potential is going to start in the same way. So don't think that this presynaptic neuron just magically had its own action potential. That action potential was subject to the same types of rules that we've talked about before. So the action potential, once it does happen, gets conducted down the length of the axon, as we've discussed before. Once it reaches the axon terminal, this opens voltage-gated calcium channels, and we release neurotransmitter into the synaptic cleft, this area in between the two cells. This neurotransmitter will bind to ligand-gated ion channels, which opens up these ion channels, and we get some type of ion flowing through. For the purpose here, we are assuming it's going to be a sodium channel, which will give us an EPSP. And then everything else after that happens exactly the way that we've talked about. The EPSP will spread. If we get to the axon hillock, what's called the axon initial segment here, and we pass threshold, we start the action potential, we open voltage-gated sodium channels and then potassium channels, and then we conduct a new action potential in this postsynaptic neuron. And then from there, we can start the whole process here again, where this postsynaptic neuron could then be considered the presynaptic neuron for when it signals to the next cell in the chain, whether that's a neuron or a muscle fiber. It just depends. So let's talk about those neurotransmitters themselves, because similar to hormones, there were dozens upon dozens of different types of hormones that we had to know in Chapter 17. There are not quite as many different types of neurotransmitters, but there are several different types. So the idea here is that when a neurotransmitter, whatever type it is, when it opens a ligand-gated ion channel, the membrane potential of the postsynaptic cell is going to change in some way. We've already said that this change can happen in one of two ways. Either we get depolarization and it's an EPSP, or we get hyperpolarization and it's an IPSP. So do you remember what determines whether the membrane is depolarized or hyperpolarized? Well, it's all going to be what kind of ligand-gated ion channel was opened. If it's a sodium channel, it's going to be an EPSP. If it's a potassium channel, it's going to be an IPSP. And there are other cases as well. So you need to be able to track what kind of ion you have. Is it positive or negative, and in which direction is it moving? Is it leaving the cell or is it entering the cell? And what effect will that have on the membrane potential? Okay, second question. What determines which type of channel gets opened? Well, it's all going to be depending on the combination of neurotransmitter and channel and receptor, right? So different types of neurotransmitters, depending on what channels they bind to, are going to open sodium channels in this context or potassium channels in another context. So for that reason, it's going to be worth our time to look into what types of neurotransmitter receptor combinations could lead to an EPSP and which types could lead to an IPSP. And there are even going to be kinds that could lead to either or. It's just going to depend on context a little bit. So to this end, there are two different types of neurotransmitter receptors. We can classify them in a very simple way. The first is called an ionotropic receptor. So we've looked at this before. This is the type of ligand-gated ion channel in which the channel itself is the receptor. So in this case, you have a neurotransmitter in this kind of red hexagon shape. It binds to the channel itself. The channel is the receptor, and that binding causes the channel to open up and let an ion through. So because no signal transduction mechanism is required here, these tend to work very, very quickly. All you need is the neurotransmitter to bind. The second type is called a metabotropic receptor, and these are going to be a little bit slower, and you'll see why here in a second. Metabotropic receptors themselves, the receptor that binds to the ligand, the neurotransmitter, it is not actually the ion channel. The ion channel is a separate protein that is nearby in the plasma membrane. Metabotropic receptors, when they bind a neurotransmitter, will activate a G-protein coupled receptor mechanism. So the metabotropic receptor is a G-protein coupled receptor. When the neurotransmitter binds, we will turn on a signal transduction and second messenger cascade, and that is going to be what causes the ion channel to open. So like I said, because this is a little bit more time consuming and because we have more things that kind of have to occur before the channel opens, these are going to act slower than your ionotropic receptors. So the reason why we care about this is because different types of neurotransmitters will activate different types of receptors, so we need to be aware of that. Now, let's talk about individual neurotransmitters. By far, the most common one we are going to see more than half of the time is going to be this one right here, called acetylcholine. Many, many, many neurons in the body will release it once they're done firing off their action potential. So best examples are somatic motor neurons, which are motor neurons that signal to skeletal muscle fibers, and then parasympathetic autonomic motor neurons, which we will explore in chapter 15. So acetylcholine will bind to two different types of receptors, but they are both acetylcholine receptors. And which one it binds to will dictate the response. The first is called a nicotinic receptor. If that name seems familiar, if you're thinking, oh, that sounds a lot like nicotine, you are correct. These receptors were named because nicotine is an agonist for these receptors. So nicotinic receptors are ionotropic receptors, meaning a nicotinic receptor will act very quickly because it itself is the acetylcholine receptor. The nice thing to know about nicotinic receptors is that they will always be sodium channels, and therefore they are always going to be excitatory, meaning they will cause EPSPs every single time. The other type is called a muscarinic receptor. Similarly, the chemical muscarine is an agonist for these receptors. These are metabotropic receptors, and therefore they're going to act a little bit more slowly than their nicotinic counterparts. And there are different subclasses of muscarinic receptors, meaning one type of muscarinic receptor may be a little bit different than another. And to this end, some types of muscarinic receptors will be excitatory, meaning they will cause an EPSP, and some of them will be inhibitory, meaning they cause an IPSP. So here's your nicotinic receptor. So we already said that it is ionotropic, and you can definitely see that when it is open after binding to acetylcholine, it lets sodium right on through, and that depolarizes the cell. Here's your muscarinic receptor, which again, the muscarinic receptor is metabotropic because it binds to the acetylcholine, but it itself is not the ion channel. We have to activate a G protein coupled receptor pathway that then causes these G proteins to become activated and move over, and they open the ion channel. So look at this situation here. Is acetylcholine causing an EPSP here or an IPSP here? So definitely look at what ion is being let through here, and you should get your answer. Other than acetylcholine, another one of our common neurotransmitters will be norepinephrine. Norepinephrine activates a class of receptors other than acetylcholine receptors, so acetylcholine only binds to nicotinic and muscarinic receptors. Norepinephrine will instead bind to a different type of receptor called an adrenergic receptor, and these are always going to be metabotropic. There are no ionotropic adrenergic receptors. So it is worth mentioning that epinephrine, which we talked about in chapter 17, epinephrine will also activate these same receptors, these very same receptors, but it does not do so as a neurotransmitter. It does it as a hormone released from the adrenal medulla. So norepinephrine and epinephrine are going to be a very common topic of conversation when we start talking about the autonomic nervous system in chapter 15, particularly the sympathetic division of that. Some other neurotransmitters that you could be aware of, but we're not really going to get a chance to talk about very much. Dopamine and serotonin each have their own class of receptors that they bind to. Dopamine is a neurotransmitter that's mainly released from these places in the brain called the substantia nigra and and other places within the midbrain. Amino acids, individual amino acids like glycine and glutamate, and a modified one called gamma aminobutyric acid. They will each have their own type of receptors too, but we're not really going to talk about them too much, so don't worry. And then neuropeptides, basically just little chains of peptide, kind of like oxytocin, an antidiuretic hormone, can also act to suppress nociceptive or painful sensations by binding to ligand-gated ion channels. All right, let's talk about the distinction between excitatory versus inhibitory neurotransmitters. This is one of the major things that we care about for any given neurotransmitter. So a neurotransmitter will be considered excitatory, simply put, if it stimulates an EPSP in the postsynaptic cell. That's it. Inhibitory neurotransmitters do the opposite. They stimulate an IPSP in the postsynaptic cell. some neurotransmitters are going to be kind of either or. They will be excitatory if they cause an EPSP, or they will be inhibitory if they cause an IPSP. Some neurotransmitters are always excitatory and some of them are always inhibitory, but a good number of them are either or depending on what type of receptor they bind to. We've already seen how acetylcholine will always be excitatory when it binds to nicotinic receptors, but could be either or if it's binding to a muscarinic receptor. So the question becomes, what ultimately is going to determine whether a neurotransmitter is excitatory or inhibitory? If I asked you a question on an exam and I said, okay, this is neurotransmitter A, is it excitatory or is it inhibitory? What you might be wondering is, what information do I have to have in order to make that determination? There's one very specific piece of information you absolutely have to have if you're going to answer that question. You have to know what kind of ion is being let through the then opened ligand-gated ion channel. If you do not have that piece of information, you cannot hope to answer the question. So if I say this is neurotransmitter A, and that's all you know, what kind of neurotransmitter is it? You'd say, I don't have enough information to answer the question. Only if I tell you what type of ion is being let through are you going to be able to make that determination. The only exception to that we've talked about is if I tell you acetylcholine binds to a nicotinic receptor, you would say excitatory because you know all nicotinic receptors are going to cause EPSPs. Okay, so now that we have released neurotransmitter into the synaptic cleft and we understand it's going to bind to neurotransmitter receptors on the postsynaptic cell and cause a new graded potential, how the heck do we turn this signal off? So how do we get neurotransmitters to stop doing their thing? Similarly, we had a discussion in chapter 17, how do you get hormones to stop doing their thing? You got to get the hormone out, right? So if we're going to turn a neurotransmitter response off, we have to get rid of the neurotransmitter. So the way this typically works is that neurotransmitter that is actively in the synaptic cleft, then you got to get rid of it. And the way we do this is with enzymes that are on the extracellular side of the plasma membrane of the postsynaptic cell. In this case, with acetylcholine, we have an enzyme called acetylcholinesterase. So this is an enzyme that only works on acetylcholine. There will be other enzymes for norepinephrine and other things like that. So this enzyme will destroy acetylcholine, basically split it in half into an acetate molecule and a choline molecule, and then those can be recycled back to be reused. Other types of enzymes will simply load the neurotransmitter back into the presynaptic cell so that it can be reused again. So either way, we get neurotransmitter out of the synaptic cleft. We take the ligand away from the receptor, and the whole mechanism can be shut off. So this table here, you can see many different types of neurotransmitter and their different classes. The ones we mostly are going to be worried about are the cholinergic, which is just acetylcholine, and the biogenic amines. Mostly it's just going to be norepinephrine, epinephrine, and dopamine. All the others we're not really ever going to talk about. All right. Last thing that we want to discuss here, now that I feel like we have a pretty complete understanding of individual topics within the nervous system, let's put it all together. So we have a little bit more to say about graded potentials and action potentials before we finish up here. So you will recall that the whole purpose of a graded potential is that it exists to give you an action potential. If a graded potential is not strong enough, it doesn't give you the action potential. If it is, you will, right? So the issue is that, as we've discussed, graded potentials come in varying grades of strength. That's why we call them graded potentials. And as we've said, an insufficiently strong graded potential is not going to give us the action potential that we so desire. The first thing that we want to do here is we have to understand the relationship between the strength of a graded potential and the amount of neurotransmitter. So what I'm going to do here is I'm going to redirect you to a separate video that I will have recorded and posted on the relationship between how much neurotransmitter is in the synaptic cleft and how strong of a graded potential we get. So you may want to go ahead and take a break here and go look at that video instead. That should not be a very long video, so hopefully it's not too much of an issue for you. So go ahead and do that, and then we'll pick things up back here when you come back. So after you've had a chance to look at that video and you saw how progressively increasing the amount of neurotransmitter that is available to bind to neurotransmitter receptors, you see how we're able to get more sodium to flow into the postsynaptic cell, which produces more and more and more depolarization and gives us a better and a better shot at an action potential. So that's the relationship that we want to see there. So in that video that I posted, what I showed was more and more neurotransmitter being released from a single axon terminal. Well, that's not really realistic. So most axon terminals are going to kind of release a standard amount of neurotransmitter. Where you get stronger and stronger graded potentials is not from one axon terminal releasing more and more neurotransmitter, but rather from many axon terminals that all converge on the same postsynaptic cell. So the idea here is that just because the release of neurotransmitter from one presynaptic neuron axon terminal, just because that's not enough to give you an action potential, that doesn't mean we can't get an action potential, especially not if we have multiple axon terminals synapsing on this one postsynaptic cell here in white. So in green here are the axon terminals of neurons that will release excitatory neurotransmitters, meaning we will get EPSPs. In red here, we've got the axon terminals of neurons that are going to release inhibitory neurotransmitter, meaning we get IPSPs. So if all these neurons fire their action potentials and release neurotransmitter at about the same time, all those neurotransmitters can get to work on this postsynaptic cell at the same time. So it's like having a whole bunch of extra neurotransmitter. So the idea here is that these graded potentials, both EPSPs and IPSPs, add together. They summate, meaning if the postsynaptic cell has multiple presynaptic cells that are all signaling to it simultaneously, we just add up the graded potentials and their effects. So if you look at the diagram here and we start at resting membrane potential and we get two EPSPs coming first, you can see that those two EPSPs add up, but they don't quite get us to negative 55 to threshold. Then we get an IPSP to take us down, then an EPSP to take us back up, then an IPSP back down, and then finally a relatively strong EPSP to get us up to threshold so that we get our action potential. So something that is definitely worth being aware of here is whether we get an action potential in this context could very much depend in which order do those neurons release their neurotransmitters. If all the EPSP ones go first, then we're definitely getting an action potential. But if the IPSP ones go first, we might not get an action potential because we start off very hyperpolarized. It's worth kind of being aware of. So this is the sort of thing that kind of backs up what I showed in that video that I posted, where more and more and more EPSPs produce more and more and more excitatory neurotransmitter, opening more and more ion channels, which produces a much greater deal of depolarization, which is going to give us a better chance of reaching threshold eventually. Here you can see another case of EPSPs adding together. In this case, we have a postsynaptic cell and two presynaptic cells. If only neuron number 1 up here releases its excitatory neurotransmitter, you can see that's not enough to get us to threshold. But if both of them release their neurotransmitter at the same time, those EPSPs add together, and that ends up being sufficient to get us to threshold. So this cuts both ways, though. You can add EPSPs and IPSPs together as well. Same situation here, we have one postsynaptic cell and two presynaptic cells. Neuron number 1 releases an inhibitory neurotransmitter that will hyperpolarize the postsynaptic cell, whereas neuron number 2 releases an excitatory neurotransmitter that will depolarize it. So the idea here is that since neuron number 1 releases its neurotransmitter first, This greatly hyperpolarizes the postsynaptic membrane and makes the action potential very difficult to achieve. So that by the time the EPSP starts, we are so hyperpolarized that we're not able to get up to threshold. And you can imagine here, if we had just released that excitatory neurotransmitter first, we probably would have gotten an action potential. But what the IPSP did there is it made it more difficult to achieve our action potential. And basically what it does is it raises the bar for how strong of a stimulus it's going to take to give you an action potential. So IPSPs are not always bad. They act to desensitize a neuron and make it take a very strong stimulus to give you an action potential. Sometimes you don't want to have to respond to every single little stimulus. Sometimes having these IPSPs come in and raise the bar for what it's going to take is a good thing. All right, so that finally does it for Chapter 12. I very much thank you for your attention here. I should also have a kind of overview video posted that kind of covers generally everything from neurotransmitters to graded potentials to action potentials posted, so I definitely recommend you take a look at that. I've said enough at this point, so I will sign off, and I will see you next time for Chapter 13 on the central nervous system. Bye-bye.